All right, let's jump right into it. Got team set up. You guys are in teams. Can you guys see me? Okay, cool. Hey guys. Okay, so we're gonna go over this last portion of glycolysis. We're gonna go over a little bit of Krebs. We're gonna go over kind of like a hodgepodge of some few things for metabolic processes. Okay. So first things first, glycolysis, it occurs everywhere in the body. Okay. And gluconeogenesis, it occurs just in the liver, just in the cortex of the kidneys, right? Reason for that is that it doesn't provide us with energy. It doesn't give us any ATP. It doesn't do any of that. Um, Mainly, gluconeogenesis is just think about it as everything that happens in glycolysis is just the exact opposite. Okay. So as we see this picture here, which we'll go over some of the stuff in just a moment. That's the Krebs. Say hi, Krebs. Okay. So we got gluconeogenesis and glycolysis. All it is just one happens the opposite way of the other. Okay. Glycolysis, we start with glucose. Okay. You can have hexokinase, glucokinase. In this class, you're not going to be tested on that, so I wouldn't worry about that, the differences of those. You're going to break down. Okay. Steps 1, 3, 7, 10. Goes all the way to 10. But glycolysis is meant just to create a pyruvate. That pyruvate is going to be funneled down to here. They're going to pluck off acetyl-CoA, boom. Then it makes, I'm sorry, they're going to pluck off the CoA group. Acetyl-CoA is going to merge with the oxaloacetate. We're going to create a citrate. Okay. 1, 2, 3, 4, 4 carbons, oxaloacetate. Citrate 1, 2, 3, 4, 5, 6. Okay. That makes sense? Yeah, it's I wouldn't say not to memorize all of these, but then you would also have to memorize all of these, and then you'd have to also memorize the reverse. Yeah, as long as you understand where the enzymes are and what the enzymes do, you'll be fine and not to overthink things. So for example, so we got oxaloacetate, we got a citrate citrate synthase. Wow. So we have three enzymes that are going to be in this lesson that are very important. I'm going to jump back to them 15,000 different times. A kinase adds a phosphate group. Okay. So oxaloacetate is going to be added with an acetyl-CoA group. CoA group gets out of here. It's out. It's done. We create citrate with a citrate synthase enzyme. Okay. Water breaks the bond between these two. That's a hydrolysis. Okay. And if I'm going too fast, let me know. I can break every one of those things that I'm saying. Got it. Okay. Just to recap. So hydrolysis, it's mainly just breaking a bond using water. Okay. So it breaks the bond, CoA out. We grab the two carbons from acetyl group because acetyl's got two carbons. That's a six carbon group. Six carbons, six carbons, six carbons, five, five. I'm sorry. Whoa. Six carbons, six carbons, six carbons, five carbons, four, 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 four. And it starts over. Six carbons, five, four, 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 four. So as long as we're following the carbons, we know that we're losing a carbon. Where's that carbon usually going to be going? Just think about that for a second. We're losing the carbon group as we're going around the Krebs cycle. Yes, yes, it's being released as CO2. So we're giving credit to the plants, helping them breathe. Exactly. Exactly. So we're tossing this out. Okay. So, oh, perfect. It even has it on here too. So see how there's a CO2 that's leaving. Boom. And so 1, 2, 3, 4, 5. Oh, what's that? 1, 2, 3, 4. So we have 4 carbons right there. Okay. So we're losing a carbon, losing 2 oxygen groups as well. That's being released as we exhale. Okay. 4 carbon, 4 carbon, 4 carbon. Might be a little ridiculous to think about it, and this might be a little too far, but every time you're doing one of these, you're exhaling. It's it's not, but just think about that like that. If you're losing a carbon, it's being released as CO2, carbon dioxide. Yes, correct. Okay. Recap, glycolysis, which we'll go over in just a second, occurs everywhere. Gluconeogenesis, just the reverse. Break it down. Gluconeogenesis. Genesis creation. Gluco, glucose. Neo, creating. New. Baby. Wah wah. Okay. Pyruvate going all the way up to here, creating glucose. So this is just reversing it. Glycolysis is going, okay, we have glucose. Let's break it down. Let's make some ATP. Oh, got my dog right here. Okay, Papa. Okay, so you got glucose. Go over some of these important intermediates, and we'll go over the enzymes. While we're here, isomerase, what does that mean? Biochem, fanatics. Yep, just changing something to something else. Simple, love it. Couldn't have said it better. Okay, so let's go back to Krebs. Citrate synthase. Yep. So it's combining an oxaloacetate, that's a 4 carbon group, to make citrate. Explain that again. Yeah, the intermediate, it's cityl-CoA, cityl-CoA. You're not going to be tested on that. Exactly. Cool. So hydrolyzes it. Yep. So forming the cityl-CoA will hydrolyze it, release the CoA group. The CoA group's gone. Okay. Acetyl-CoA oxaloacetate form to make citrate. I would leave it cut and dry as that. Okay. So enzymes give I'm trying to think how to explain this. Enzymes give us clues to what's being made and how it's being made. Okay. So if the enzyme is citrate synthase, that means it's adding what? Well, yeah, it's just breaking the bond using water. Yeah. But what else? Bingo. Got it. Good job. Okay. Any questions? Any questions on any of this? Well, probably a lot, but any questions on that so far? Okay. Let's take a step back. Okay. So kinases add a phosphate group from ATP. Okay. Phosphorylase adds a phosphate group to free phosphate, ADP. Okay. ATP is so negatively charged, it repels each other when it pulls a phosphate group from there, and that produces energy. So think about that as, like I was telling you, yeah, like the last class. So there'd be 2 magnets, negative and negative. We would have to shove them together to hold them together. Now imagine when those magnets are released, boom, all of that energy is going to be released. That's going to be fed into all of our biological systems. Correct, exactly. That's how ATP works. Hmm. Yeah, because resonance, exactly. But I would think about it more as their phosphate groups are negatively charged, like 2 groups, right? So they do not want to be near each other. So they force that ATP to go, we need to give this phosphate group. We do not. They're trying to, they got, yeah, Step Brothers. Think about Step Brothers, the movie Step Brothers. They're trying to kick him out, kick out Dale. Tell him to get a job, right? Trying to kick out Will Ferrell. Exactly. Okay. Phosphorylase still adds a phosphate group, but it's not forcing it. Okay. We got one other. It's a phosphatase that removes it. You might have it. Yeah, I like that one better. There you go. Phosphatase removes a phosphate group. Could you think of any scenarios where we would use a phosphatase as opposed to a kinase? Anyone? Anyone? Yes. I see how that could be confusing. I see that. Okay. I can go a little bit more into depth than that. My apologies. Of course. Got it. Yeah, you're thinking the right way. Okay. So a phosphatase removes a phosphate. Do you want it? Okay. Yeah, I can explain that a little bit differently, of course. So a phospho, let's see, a kinase group, phosphoglycerate kinase. Okay, that you're going to add um, it's going to transfer a phosphate group into there. No, I'll pull it up for you. Here we go. And I might have to pull up a better picture. Okay, so this is really just a structure. So you'll see there's two phosphate groups on it. Okay, on this 1,3 biphosphoglycerate. Okay, it's going to turn to 3-phosphoglycerate. Why? Anyone? Anyone? Yes. Yes. Those are all good questions. Hmm. Well, no, no, no. It's gonna okay. And for everyone on Zoom, Teams, you guys are asking great questions. If you guys on here on Zoom want to chime in, um, yeah. So it's going to give an ATP group to ADP. Exactly. It's going to give it to ADP. So a kinase is going to transfer a phosphate group to ADP to make ATP. Okay. Let's jump back to it. You guys see my mouse? Boom, boom, boom. Okay. Okay. Anyone? 1,3 biphosphogycerate. We need to have ADP plus a proton, so plus the H plus group. Mhm. It's going to turn to 3-phosphoglycerate because we're losing a phosphate group. There we go. Good job. Good job, guys. A lot of you guys are getting it. So we're plucking a phosphate group onto it. Where is it going? It's going to ATP. They're building ATP. That's the whole reason for why we're doing any of this. Well, and also remember, we got to get back to the plants, right? CO2 out. I say that jokingly, but that's a big portion of Krebs cycle that's forgotten. If you're counting the carbs, the carbons, and you can follow the CO2 out, get back to the plants. I always think about that. Okay, good job. So we lose a phosphate group on the one side, on the one carbon, right? Does that make sense? If you guys want me to draw it out, I can. It's gonna look really bad though. I'll draw it out though. Okay, thumbs up, thumbs down. Who wants me to draw it out? Anyone? Am I talking to myself? Okay. Well, I gotta figure out how to do so 1,3. So I'm drawing this. Okay. Everyone focus. Don't write this down. Just look. Okay. See, we got a phosphate group. Okay. I'm not drawing a phosphate group. Okay. It's connected to an oxygen. Okay. Is everyone following? Oxygen, carbon, double bonded to an O. Horribly drawn, but that's okay. You guys will be fine. OH and H. Okay. Everyone following? Good. Okay. See? And then it'd be what is CH2 phosphate group. PO3 2 minus. Same thing here. PO3 2 minus. Yeah, I'll write it out. Yeah, PO3 2. Yeah, it's just phosphate. Exactly. Cool. So when 1,3 goes to 3, we're plucking off the carbon 1. 1, 2, 3. Does that make sense? Don't just say yes. Does that make sense? Yes? Okay, good. So we're plucking this group off. Oxygen's still hanging there. Okay. Now, what are we going to do with that bond? It's become a double bond. It's become a negative group. Let me write this in a different color. And I'll eventually get better at drawing on the computer. I'm so not used to it. Okay, cool. Double bond, because it loses the bond to phosphate. Becomes negatively charged. Double bond. Now, this phosphate group goes to an ADP to form ATP. Does that make sense? Okay. That tells us when we see kinase that this is happening. That is the enzyme. Exactly. I gotcha. Great question. Great. I love that. Great question. A kinase can add or it could take away. It could transfer. Okay. I wouldn't think too much of it. Just think of when you see that kinase, you're adding a phosphate group because this is being added to a DP. ADP was over here, and then it has H plus proton. It will have that. Memorize that. Then it's gonna bond with one more ATP. Boom. Got it? I don't want to spend too much time on that, but make sure you guys know that. Okay. I'm reversing all this to clear all drawings. Oh, there you go. Look at that. Getting better at these. Any questions? Yes, 1,3 BPG is now 3 PG, 3 phosphoglycerate. Okay. Enzyme used is phosphoglycerate kinase. Okay, let's break it down. Kinase, phosphate, mhm, glycerate, glycer, glycerizy, got it. Biphosphate, 2 phosphate groups. There's the 1 and the 2 that I drew. Okay. Yeah, it's a glycerol. Glycerol is for fatty acids. You guys aren't being tested on that. You're fine. That's why I'm just skimming over that. Questions, questions. Okay. So why can glycolysis proceed in the absence of oxygen? Think about that for a second. And you can put your answers in the chat box if you feel comfortable. Not, that's fine. But just think about it for a second. Even if you don't know the answer, why do you think that glycolysis would proceed in the absence of oxygen? Think about where glycolysis occurs. Um, where does glycolysis occur? Hmm. Yeah, that is all. Where does the electron transport chain occur? Okay. Where does the Krebs occur? Good job. Okay. So if we need oxygen, I'll show you this so you can see where my mind's thinking. So without oxygen, glycolysis will go through another process to still go. It doesn't need oxygen because it's in the cytosol. Okay. But without oxygen, the electron transport chain will get shut down. Okay. The electron ETC. Yeah, I can go over that simply, but it's just a place where it's just a place where NAD and FADH2 are just converted to ATP. That's it. There's different complexes, so the longer you're in the cycle, the more ATP is produced. So, good question. Good question. Okay. I'll answer that after, because this class doesn't go too much into lactate fermentation cycle. But I'm just explaining it, what would happen with the oxygen. Exactly. Okay. So without oxygen, glycolysis will go through another process. So I'll turn pyruvate to lactate. See here. Okay. So here, you'll have a little step where it turns to lactate. Now, mhm, lactate can now turn NADH to NAD plus. It's lactate fermentation cycle. Okay. So without oxygen, ETC will be shut down. NAD cannot be oxidized back to NAD plus. Okay, remember that. Also, yeah, good question. Where does pyruvate come from? Glycolysis. Good job. And it's the end product of breaking down glucose in where? Cytosol. Good job. So glucose, it now that we have glucose, it'll turn it into how many pyruvates? Okay. Hi guys. Does everyone have an answer? Yeah, these concepts will be tested on the final for sure. I would know these, at least the idea of them. You're learning it all here. You're going to learn it all here. You're fine. That's fine. Yeah, it's biochem. There's always something to learn. hmm You may forget one thing and learn 10 things. So I I would say before learning all the enzyme substrates and like all the intermediates, I would understand why everything's happening. Like that's always started with the kinases. Um, Correct. Yeah. So if you're given a question, you can know, okay, well, this is a kinase. I know what that enzyme does. Okay. Well, this is going to add a phosphate group somewhere. So, okay, where's that coming from? You know where you can look at the intermediate and go, okay, well, this is probably in glycolysis. Oh, this is probably in Krebs. Okay. Got it. Cool. Okay. Ligand gated channel. What is that? It's receptor class. Cool. Pore channel. Good. Regulates ion movement. Good. Okay. What does it do? Okay, so it opens and closes. It's not just opening. It can open or close when there's a ligand. So think about it as um, think about it as like a neurotransmitter will bind. Okay, it'll change the shape, and then it'll allow an Na plus, K plus, Ca2 plus, Cl minus. It'll allow those to rush inside the membrane. Does everyone understand that concept? Yes. Okay. Why is that so important? Can those ions enter the membrane? Do you think so? Okay. Why can they enter the membrane? Yeah, they can't. They can't enter the membrane. They need help. Okay. So let's backpedal for a second. So to pass the cell membrane, you need to be a small non-polar group. Okay. So let's think of some non-polar groups. So you get what? Steroids, testosterone, estrogen, cortisol, aldosterone, progesterone, anything that's going to branch from from a cholesterol group. Okay. CO2, NO2, sorry, CO2, N2, O2, fatty acids, the vitamins are lipid soluble. You guys are on it. On it. I love it. Cool. Okay. So what cannot pass? Charged ions. You said it right away. Charged ions. We got Na plus, K plus, Cl minus, Cl. I'm sorry, calcium 2 plus. Glucose, proteins. Mhm. Well, it needs to transport it across. If it mixes with H2O, then it won't cross the membrane on its own. Insulin. Love that answer. Insulin. Good job. You're reading ahead over here. Don't look at that question. Okay. So let's take a further step back. So there's three classes of receptors, okay? And you will be tested on these. GPCRs, what is that? Yeah, just makes a protein cascade inside the cell. So there's a 7 transmembrane receptor. So it goes in and out 7 times. 1, 2, 3, 4, 5, 6, 7. Okay. A ligand will bind outside. It'll activate a G protein. Oh, we got 10 minutes left, guys. Exactly 10 minutes. Let's rush through it, okay? Yes. It'll activate a G protein inside the cell. There's a complex that'll happen through there. If you want me to explain it, very high yield. Very high yield, yes. Okay. Activates a protein inside. Protein phosphorylation cascade is activated. Okay. Secondary messengers, you got what? Name them. As you know these off the top of your head. CAMP, IP3, DAG. CAMP, IP3, DAG. Secondary messengers. So when you see those, just think GPCRs. Got it. Yeah, I would. I would ask the question, have you looked, do you understand how this process works, the GPCRs? And I always put this at the end of it because it's going to get the best pictures. Yep, cool. Okay, so let's look at it. So it's loading here. Yeah. Okay, so there's the G protein, 7 transmembrane receptors. Okay. Ligand binds outside the cell. That could be whatever, whatever, right? Hormone, something. Activates the G protein inside. Okay. You guys see this? Protein phosphorylation cascade is activated. Okay. CAMP. Got it? Protein phosphorylation cascade activated. Hmm. So that's when your secondary messengers, CAMP. Okay. And this signaling is so fast. It's ridiculously fast. Okay, this actually might be a better picture, so I can explain the portions inside of it. By the way, Jack Weston, y'all, if you have heard of it, haven't heard of it, amazing. I can't say enough about Jack Weston. They're great, great pictures. Okay, look, ligand. Okay, anyone. First messenger binds. Okay, this is the same thing. 7 transmembrane. Ligand binds outside, activates GPCR, G coupled protein, G protein coupled receptor. Okay, GDP turns to GTP. Okay, binds the enzyme. Inactive second messenger, active second messenger. Cellular response. Cytoplasm. Got it? Yeah, just think epinephrine, glucagon. Yeah, you guys got it? Cool. Ions, channel receptors. They have a picture here. I mean, it's the same thing. It's just a ligand binding. Channel will open. Think of a channel as like these little. They're closed, so that they're trying to bump it through here, and then now it opens. So now you'll see sodium go in. You'll see potassium go out. You see that could be like a GABA receptor, or let's say like a chloride channel. There could be an acetylcholine receptor for some kind of, yeah. There's a lot of different things. But ion channel receptors open, so ions, that means charged ions cannot enter the membrane without an ion channel. Does that make sense? Drive that into your brain. I would say it 3 times. Got it? Got it. Okay, good. Huge, 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 huge. High yield. Incredibly high yield. Yes. Okay, cool. Third, enzyme receptor, nope, enzyme receptor, enzyme linked receptor, sorry. That's when there's a reaction that triggers in the cell. So there's a ligand that will a ligand that'll bind. So same thing. So let's look at this picture. I can find a better picture for you. But then there's a receptor that'll dimerize. Okay, it means they'll move apart, and then that enzyme activity is turned on. Okay, think about receptor tyrosine kinase. So that will need, if it's a receptor that needs a tyrosine, that means it's a receptor with just a bunch of tyrosines on it. Okay, think about these two walls. It'll start a process inside our body from this enzymatic reaction. Okay. Those are very slow though. They're very slow to happen, but they're very long lasting. Okay. Yeah. Insulin. Insulin. This is insulin. Okay. Insulin will bind to the tyrosine kinase. The dimerization will occur. So bringing both of their beta subunits. I might have to pull up a picture, but their beta subunits to pull these subunits together. You'll autophosphorylate, meaning that it phosphorylates on its own. Okay. Good question. Okay. Yeah. So let's pull that up then. I can see if they have like one for tyrosine kinase. You'll see these beta subunits will phosphorylate each other. Um, uh, Crappy picture, but it does the job. Hopefully you guys can see it. Yeah, I'm talking about this one right here. Where to go? Well, yeah, I guess this is the concept. It's loading, so it's gonna move away in a second. Okay, what the heck is going on? Yeah, got it. Okay, well, here, this is good enough. So there's a signal molecule. They're inactive. They dimerize each other. Means they autophosphorylate each other. Okay, it activates that kinase domain. The phosphate group will be added to the tyrosine residue, PO43 minus. The insulin receptor substrate will now bind to it'll be called a phosphotyrosine. Think about it like a magnet. Okay, that's where this is pulled together. Okay, that's turned on now. Now everything will happen. Yeah, um, I would say it's very low yield for that. AKT pathway, PI3K. That's just saying you're, yeah, no, great question, great question. It's for metabolism. So, what he's asking is PI3K, AKT pathway. Is that turn it on the same way? No, it's a GLUT4 that's moved to a membrane. So now there's a glucose that's there. Now glucose can uptake, so you can see, glycogen synthesis. You'll see that it decreases gluconeogenesis. So if it's glycogen synthesis, it's not gluconeogenesis. You'll do one or the other. Does that make sense? Okay, got it. You see a picture of that? Okay, let's go. That's okay. So for here, making glucose, breaking that down to pyruvate, why would we be breaking pyruvate down to make glucose? So if one goes up, well, the other one goes down. Exactly. Okay. MAPK, that's just for protein synthesis. This would be a better use of your time, glycolysis. Yes. Okay. So glycolysis occurs everywhere, gluconeogenesis. You'll have that process will start when you're starving. Okay. So it'll grab glucose, it'll dump it in the blood. Okay. That's about all the time we have left. I have some more here, some more questions here, but I would leave you guys with this. Just think about when a phosphate's pulled off, right? There's ATP and ADP. These kinases right here, oh, we have it here somewhere. These kinases are going to tell you where the process is going in a Krebs cycle or in glycolysis. Okay, very, very, very high yield to understand that. Okay. Um, yeah. Any questions? Glucokinase, hexokinase. One is just to depend on the affinity for low kcat, high kcat. Um, I believe gluco, yeah, glucokinase is a high kcat. So that means it's not that great at getting glucose, but if there's a lot of glucose, it can make things happen very fast. Um, Steps 1, step 3, we put ATP in. Step 7 and 10, we get 2 ATP out. Got it. So yeah, net is 2 ATP for glycolysis. Good job. Got it. Cool. Well, you guys did good today. We jumped through it. We jumped through a lot of topics very quickly. But if you guys understand that at least, just what we just went over, you guys will do well on the final. Yeah. Just go over what we talked about. Let's see if there's that's a good question here. Let me pull this back up. Okay. Yeah, we can get over that. That's fine. The the time refreshed, so it's not gonna kick us out. Okay. Okay. So what do you think? So how to turn insulin signaling pathway off? So how do we do that? How do you think? Okay. Well, what do you think? Well, yeah, a phosphatase will remove a phosphate from activated signaling proteins. Yeah. So, okay. So when insulin binds, yeah, yeah, you have it backwards. So when insulin binds, the receptor autophosphorylates. That was that picture that we were going through. Okay. And then it'll have those other downstream effects for, AKT, MAP, or MAPK. Okay. So phosphorylation would be on. Now, if that pathway was shut down, you would remove a phosphate group from the insulin receptor, that IRS, whatever you want to call it, right? Yeah, that would dephosphorylate it, making it inactive. So basically, insulin would be degraded. It would have a smaller half-life. It would be shortened in the blood. Okay, those GLUT4 vesicles would be pulled back into the cell hmm when signaling stops. So yeah, yeah, the pathway would be shut down. So activation of a PKA would lead to phosphorylation of target proteins. Okay, phosphorylation. Remember, serine. What are the other two? Threonine, tyrosine. Good job. Okay, so GPCR, G subunit, it will have a adenylate cyclase activation. Okay. Okay. Yeah, yeah, that's all could be in the signal transduction pathway. So so think about it. If a G subunit can't hydrolyze a GTP, I think there's like a mutation or something that's going on. And that GTP never switches to a GDP, triphosphate to a diphosphate. Simple terms, right? T never goes to a D. It never inactivates, and that G protein is always on, always, 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 always on. It's permanently on. Okay. So if that pathway is active for too long, then you know, you can see that the G protein receptor is still signaling, even if there's no ligand that's binded on top of it. Okay. So if there's an excess second messenger, it's going to create too much camp. Okay. Exactly. So if we're creating too much camp, we're going to have a ton of ion loss, a ton of water loss. Okay. So if the G subunit cannot hydrolyze GTP, then this signal will never be shut off because it has to hydrolyze it to turn it off. It means you have to turn the water faucet off. So if the water faucet's constantly on, there's water constantly coming out. That means the bodies keep going, hey, we need more secondary messenger, more, 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 more. That means it's preventing GTP hydrolysis because it's locked on. hmm Cholera poisoning, cholera toxin. Exactly. Boom. Good job. You nailed that. Yeah. So, yeah, it just means adenylate cyclase will stay on. camp through the roof. Cell is pumping out ions. So it's pumping out, yes, exactly. It's pumping out Cl minus. Okay. It's pumping out Cl minus beyond this class, right? But if it's pumping out Cl minus, water is going to follow. Again. We'll go over. I'm just showing you this. You're not gonna be tested on this, but this concept you are, but not for the renal system. But just you're asking great questions. Oh, you are? Okay, cool. Yeah, yeah. This will apply to that. Absolutely. Very cool. Yeah. So PCT, boom, 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 boom. So as we can see this picture, the osmotic gradient goes as we go down, the higher the osmolarity. As we're going up, okay, we're seeing reabsorbing Na plus, K plus, right? Why am I bringing this up? Yes, because if we're bumping Cl minus out of this thing, water's going to follow. Water's going to follow. Yeah, and that's how you're going to get diarrhea, cholera poisoning, cholera toxic. Yeah, that's all from this. So you just learned something. Yeah, so massive camp build up, tons of ion loss. Na plus is out. K plus is out. Yes. Cl minus is out. Water is going to follow those ions. As we're dumping all that into there, massive diarrhea. Taco Bell. Okay. This is like instant Taco Bell. Massive camp buildup. Okay. Does that make sense? Okay. Yeah, it's a confusing concept, but I would try to take a class in your like the endocrine system. I would learn about that. That's where you're going to get a lot of this. This is well beyond biochemistry, like an endocrinology class. Yeah, I would sign up for that immediately. Okay, got it. Yeah, it's not going to be tested here, but at least now you know that concept is tested here. Yes. Okay. Okay. Any other questions before we head out? Yeah, if a GABA receptor is bound, then it's going to open up for chlorine channels. Um, voltage gated channels, those are the sodium, so Na plus, K plus channels. Um, yeah, that's for the nervous system. For creating an action potential, or enough sodium is in the is flooded, then action potential will happen. And once an action potential happens, it's an all or nothing. And that'll signal, okay, boom, we got to go. So the cell will depolarize. Depolarize meaning less negative. And then eventually when it reaches the action potential, you'll close those channels for the Na, you'll open them for potassium. And then that's how we repolarize. Got it. Yeah, it's beyond this class, but you did great. Okay. Bye guys. Have a good one. Good luck with your final. Good luck. You guys are gonna do great. See ya.